Good morning, everybody, and welcome to the Infectious Diseases Friday morning session. We have with us Kim Steegen, who is a scientist who works in our HIV resistance lab. And it's quite difficult to understand what the purpose of resistance testing is, how it's done, how to interpret it. And Kim's had many phone calls from me over the years, asking what I've seen the results that I've received. And I think it's really important to understand, especially when we're in that kind of mindset where, oh, Dolutegravir is a magic drug and it works for everything and no one's ever resistant anymore. And I think we need to understand that that's not the case, and we need to see what the future might be bringing for us.
Okay. Good morning, everyone. I just want to check if those online can hear me.
Yeah, they can.
Oh, okay, perfect. So good morning, everyone. My name is Kim. I've been running the HIV drug resistance lab for the last 12 years. I'm also the chair of the NHLS drug resistance committee. So we kind of oversee the 5 labs in the country that do drug resistance testing. And I'm gonna really start from the basics. So I'm actually not gonna focus too much on how to interpret the results. So I'm not going to go through cases. If that is something that you would like me to do, that's fine. But it's very hard to put whatever Sarah asked me in like 50 minutes. So I'm going to do my best. So I'm going to start with the principles of drug resistance. Let me see if I can advance here now. No, I can't. Sorry. Yeah, it doesn't fit on my screen. I can't see. So I see something different. Oh, there we go. Yeah. There we go. So like I said, I'm just going to talk about the principles of resistance, how the virus changes and why we actually do see resistance, and the drivers of resistance. So which factors from the patient, from the treatment, from the environment impact the chances of having resistance. Then I'm going to switch gears and go to the lab side and actually try to explain in like maybe 8 slides how we do it in the lab. It's a very complicated test that takes multiple days, and that's why you can't get a day like a result like a viral load result. And then finally, I've put some slides together on what we know for now on Dolutegravir resistance, and I'll also share something that we presented at the latest resistance conference on the prevalence of DTG resistance that we are seeing at the NHLS, which is a bit scary. So I might end on a bit of a downer, but I'm going to try to guide you through it. So obviously, before we start on resistance, we need to understand that we have different drug classes. Each of these drug classes target different stages in the replication cycle. Let me see if I can put my pointer on. Okay, here we go. So obviously there are entry and fusion inhibitors that will inhibit the initial stages. There are reverse transcriptase inhibitors, which are NRTIs and NNRTIs. Then we have integrase inhibitors that inhibit the integration of the viral genome into the host genome. We have protease inhibitors. Those ones will inhibit once you have your polyproteins, so the translation from RNA into your proteins, it will cleave them into functional proteins, and that process is prevented by protease inhibitors. And then there are also some maturation inhibitors. And as you know, we tend to treat patients with at least 2 different classes, and the reason for that is if you use a single class, the chances of developing resistance is much higher. So that's the main reason behind it. You do need to remember that all available drug classes, including Dolutegravir, there is a risk of developing resistance. Some of them might have a lower barrier to resistance, some of them might have a higher barrier to resistance, and I'll come back to how we determine the barrier to resistance. But you do need to take into account if patients are not taking their treatment well, and as long as there is viral replication, there is a chance of developing resistance. If there is no viral replication, the virus cannot change itself, so then you don't develop resistance. Okay. So why is resistance such an issue in HIV? So HIV has a very high genetic diversity. First of all, there is a production of about 1 to 2 billion viruses per day in a single patient. And because the viral reverse transcriptase doesn't have what we call a proofreading mechanism. So when it does the reverse transcription, it's not going to check if it has made any mistakes, and it's not going to correct those mistakes. Okay, so because of that, there is on average one random mutation, so one random change on a nucleotide level per virus that is being produced. If you then multiply that by 10 billion viruses, obviously you can generate a whole lot of viruses that look very similar, but are not 100% identical. When we talk about one random mutation, that does not mean that each of these changes translate into resistance. Some of those changes don't mean anything to the virus. Some of those changes may change an amino acid translation, but it doesn't have an impact on the function of the protein. So there are different gradients, if you want, of the mutations and what they mean. So if you think about a patient, you need to think this is in a very simplistic way. A patient that is not on treatment and has viral replication has a lot of viruses that look very similar but are not identical. Okay, so as you can see, these different symbols represent a single mutation. So in this case, this mutation is present in all the viruses. Okay, however, if you look at the purple cross, it's only present in 1 out of 10 viruses, for example. And this is what we call minor variants. So when we do drug resistance testing, we do something called population sequencing, which means that we are actually going to determine the sequence of the whole viral population. This means that the mutations that are present in all the viruses, we will definitely pick up. But that one that is only present in maybe 1% of the viruses, it is likely that we will not pick it up with our conventional sequencing methods. Okay, and I'll come back to why this is important at a later stage. So if we put this in a graphic diagram, if you see that this is your viral population, right? The green ones are the susceptible viruses, so your wild types, so there are no resistance mutations. The red one is one with a resistance mutation. And then the other colors in between are your variants. So they have mutations, but they don't impact on resistance. Okay. So if we then start treatment, then obviously the viral replication of the wild type is being suppressed because the patient is taking drugs. However, if there is one with a resistant mutation, that virus has the advantage and will continue to replicate even though the patient is under drug, or the virus is under drug pressure. This means that over time, your viral population will change to most of them having the resistant virus, because that has the replication advantage. If you would stop treatment, which is not recommended, initially your resistant virus will continue to replicate because that's the virus that is in the population the most. But after a while, your wild types, because remember now your drug pressure is gone and your wild type viruses are still there. So every virus is integrated into the human genome. So at any given stage, that virus can be transcribed and can be reactivated if you want. So if you wait long enough after drug interruption, you're going to have a mix between your wild type and your resistant ones. So the reason why this picture is very important is it is very important to know when to do resistance testing. Okay, so if you do resistance testing here, you're most likely going to pick up your wild type. This is why it's so difficult to pick up what we call transmitted drug resistance, unless we know that the patient is recently infected, which we all know is very difficult to determine, right? If you do a resistance testing when the patient is on treatment, that's when you're going to pick up your mutations. So that's the important point. If the patient comes to you and says, look, I haven't been taking my treatment well for the last 6 months, it might not be useful to do resistance testing because you might pick up more wild type and you might even miss the resistant mutations. Okay, so there is no point in doing a resistance test in somebody who has told you that they're not taking treatment. Okay. Oh, and then finally, just keep in mind that all the resistance mutations that have developed over time may be archived and may be reactivated at any given stage, depending on the situation that the patient is in. So what are the factors that influence persistence and reversion of mutations? So remember, when the virus does replicate, the mistakes that are made are not always wild type to mutant. It can also be mutant back to wild type because it's random changes, right? So obviously drug pressure is a very important point in terms of persistence of mutation. So as I said before, when the drug pressure is taken away, in many cases, your wild type is going to take over. Okay. Then in addition to that, you need to take into account viral fitness. Some mutations cripple the virus. Examples of that is the M184V, the K65R, and these are mutations that are actually not too bad to have. And that's one of the reasons why TreatyC, FTC is being recycled in all the different regimens, because we want to maintain the M184V mutation so that the general population, viral population, is crippled so that the other drugs have a better chance of actually fully suppressing the population that you still have. Okay. However, for those mutations, if you take the drug pressure away, those mutations are the quickest to disappear. And when I say disappear, they're not fully disappearing. They're just not appearing in your resistance result. Okay. So they are still there, but it's not the actively replicating virus. Okay. Same for K65R. If you, on the other hand, have the K103N, an NNRTI mutation, it doesn't impact on viral fitness. And this is exactly why we sometimes see patients that have not been exposed to efavirenz or nevirapine for years on end, but when we do a resistance test, we still see the K103N mutation. Okay, and it's because that virus is not affected, the replication activity is not affected by that mutation, so there is no reason for that virus to disappear. Okay. And then there are a whole host of unknown and patient-specific factors. The K103N mutation, in some patients, it stays for years on end, and in some patients, it disappears after a couple of months, and we don't exactly know why. So just keeping in mind, again, any mutation that has ever emerged will always be archived and has a potential to take over the viral population. So this is where it gets extremely important when you look at a resistance result, for example, in a patient that has been exposed to multiple regimens, but is now, for example, on a PI-based regimen, and you only have this resistance result. You always need to take the previous drug exposure into account. And you, for example, if you get that result from a PI failure, there is no K103N mutation. You shouldn't switch the patient back to efavirenz, obviously, because the patient has been exposed to efavirenz in the past, and it's very likely that that mutation is in the background somewhere. And believe you me, we have seen cases where clinicians or healthcare workers switch a PI-based failure back to efavirenz because they didn't see the K103N mutation. But then obviously that patient is going to fail very quickly again on that regimen. That was before the DTG area, by the way. So now I'm going to go through a couple of drivers of resistance. So I'm going to talk about the virus itself, the drug, the patients, the clinic, and then also the national program. So first thing that comes to mind, does subtype matter? Technically, each drug should be as efficient to each subtype. The difference is HIV-1 and HIV-2. There are a couple of drugs that don't work well for HIV-2. But because HIV-2 is not really a problem in South Africa, it's not that relevant. Even though there is no known difference in the impact that a drug has on a specific subtype, there is, however, it is known that some mutations appear quicker in some subtypes compared to others. And an example is the K65R mutation. So it's selected by tenofovir when, for those that have been in the field long enough, when the switch happened from D4T to tenofovir, most of the trials that had been done were done in the West, where subtype B is most common. And when tenofovir was used, K65R was not common at all. Then all of a sudden, we start rolling out tenofovir in sub-Saharan Africa. And the first thing we see is K65R left, right, and center. I mean, like 60 to 70% of the patients failing TEE at the time would have a K65R. So everybody was like, oh no, tenofovir doesn't work for subtype C. No, that's not the case. If you're taking your treatment well, tenofovir is working just as well for subtype B as subtype C. The difference is that the position where K65R is in a subtype B sequence, you need 2 nucleotides to be changed, whereas in a subtype C sequence, you only need one nucleotide to be changed. So the chances of that random mutation happening at the one position in subtype C is much more likely than the two next to each other in subtype B. And that is why we see more K65R. Luckily, K65R is also a crippling mutation, so it wasn't as bad as what some people initially thought. Okay. Then if you look at the viral replication, so the orange line is a drug-sensitive virus, so no mutation. So when you start, you have a very high viral load, so a lot of viral replication. As your drug concentration to the right increases, initially the virus will replicate and then it will go down. The slope of this graph is determined by the type of drug that you're taking. And for example, with Dolutegravir, this will be very steep. With other drugs, this might take quite a bit longer before you actually go down to a suppressed level. Okay. If you, however, start with a resistant virus, usually your baseline viral load is quite a bit lower. And that's because there are mutations present, and some mutations might have an impact on viral fitness. So they might not have a very good replicating virus. But in general, it takes much longer, if at all, to actually suppress that virus that already has resistance. Okay. Then talking about fitness. So this graph represents a lab experiment where they cultured virus and they added lamivudine or 3TC to the viruses to see how the viruses would react. Again, in the very beginning, you had a high viral load, so you had a lot of virus replicating. Then when you add lamivudine, the viral load would go down and very quickly M184V would come up, okay? But because this is a crippling virus, even though the viral load goes back up, it never reaches the initial baseline viral load. And the difference between the baseline viral load and the viral load over here is your loss of fitness. And this is exactly, as I explained, why we want to maintain M184V by recycling 3TC or FTC, so that we have a loss of fitness in the viral population, so that it's easier for the other drugs to actually start from a lower baseline to then finally suppress the viral replication. Then moving on to the patient, and you guys know much more about this than me, but obviously poor treatment adherence is the first thing that comes to mind. If you don't take your drugs well, you have a very high chance of developing resistance. However, there are differences. So in this graph at the bottom, you have your percentage of adherence going from 100% to less than 60%. And then here you have the rate that they actually develop resistance. So if you have 100% adherence, your chances of developing resistance is relatively low. Okay. However, if you drop your adherence up to like 70%, at this point, 70 to 79%, that's where you have the highest chance of developing resistance. Those that take less than this, the chances of developing resistance goes down again. So if you're not taking your drugs at all, obviously you're not going to develop resistance, because the wild type virus, which is the strongest virus that there is does not have any reason to maintain the mutations that it might select. Okay. And this is one of the points where I might disagree with the current guidelines, where they actually look for patients that are at least 80% adherence and only then do drug resistance testing. I think they are missing out on like a number of patients that are in that 60 to 80%. And I think those ones are also at very high risk for developing resistance. Okay. Then barriers to treatment access, cost, obviously, if patients have to pay for their treatment, travel distance, transport, parental consent for younger patients. So if they need to take medication, but they haven't been disclosed that they're taking it because they're HIV positive, how will an adolescent actually stick to taking their treatment? And then finally, obviously, stigma and discrimination. If you live in an environment where you cannot disclose for whatever reason, it's going to be much harder to stick to your treatment regimen and take your medication all the time. Having said that, missing a dose is very human. I mean, if any of us needs to take an antibiotic course for 10 days, how many of us actually do take it for the 10 days as we should? For those who are taking the pill, how many of us miss a dose just because you forget? And it's the same with HIV treatment. Okay. Then switching to the drug regimens, the potency is very important. And that's why we have now switched to dolutegravir. Dolutegravir is an extremely potent drug, which means that the viral load goes down very quickly. Okay, so the quicker you can reduce the viral load, the less the chances that the virus will develop resistance. If it takes 6 months for you to suppress the virus has 6 months to make mistakes and to potentially select for mutations. Okay. Drug-drug interactions, you know this better than me. The main one is obviously TB co-infection, but I'm sure there are other drugs that I'm not aware of that have impact on the HIV drugs, and you need to either change the drug or adjust the dose. And then poor tolerability and toxicity. So even though if you have a patient that is religiously taking their medication, if they have diarrhea or vomiting or whatever else, obviously the drug is swallowed, but the drug concentration in the body is not high enough to actually suppress the viral replication. So that's definitely something to take into account, because that might be a reason why they develop resistance. And then a low genetic barrier. So the genetic barrier is defined by the number of mutations that is required to develop high-level resistance to a particular drug. So if we think about the NNRTIs and TTC, for example, you only need one mutation and immediately you have high-level resistance. Like there is almost no efficacy of that drug left when you have that mutation. So examples are M184V, K103N, and so on. Okay. If we, however, look at a high genetic barrier, it means that if you have a single mutation, you will lose a little bit of the efficacy of the drug. If you have a second mutation, you will lose a little bit more, but only if you have 3 mutations, you will actually get to high-level resistance. And those examples are AZT, RPIs, atazanavir and lopinavir, and the second generation INSTIs. Okay, so for dolutegravir, we assume that you need at least 2 mutations to have high-level resistance. However, there are like a handful of exceptions. So I think the genetic barrier of dolutegravir is a little bit lower than we initially anticipated. And again, it is when they talk about this genetic barrier, it's all derived from lab experiments. They culture the virus in the presence of drugs and they see which mutations are being selected by the virus. But obviously, if we then put the drugs into the patients, it might look slightly different from what we've seen in the lab. Okay. So if you look at this graph, and it's a bit of an outdated graph, here are the different drugs that are available. And then you have the potency. So that is linked to the estimated log change in viral loads, so the log drop. And then you have the genetic barrier to resistance. So the higher this number, the more mutations you need to have high-level resistance. So obviously, we are interested in drugs in the upper right quadrant, dolutegravir, lopinavir, darunavir. Okay. So those ones are very potent, so a very quick drop when you start taking the drugs, and you need multiple mutations before you actually have high-level resistance. If you look at raltegravir, for example, which was our third-line go-to drug for a while, it does have a decent potency, but it doesn't have a very high genetic barrier. So that is why many patients that are on raltegravir and were not taking it very well, they actually relatively quickly develop resistance. So if you see any patient that is still on raltegravir, please, if they're not suppressed, please do a resistance test. If they are suppressed, switch them to DTG. Because also exposure to raltegravir gives a higher chance of developing DTG resistance because there is some overlap between the mutation profiles. Okay. Then healthcare facilities. Interrupted drug supply. So obviously stock out leads to poor adherence, non-adherence, and increases the risk for resistance. Suboptimal treatment adherence support, which also goes together with stigma and disclosure. In some people, SMS reminders works. In other, treatment buddies help. Some like speaking to others in adherence clubs. So, I mean, there have been a lot of social studies done on this, which is not my field of expertise. And then poor treatment retentions. People that are not adhering to their clinic visits are usually the ones that are struggling. They are usually the ones, because they're not coming to the clinic, we can't do viral loads on them. So we actually don't know how they're doing. And there has been a lot of talk now in reaching the 95, 95, 95 that we're very good at dealing with the people that are at the facilities, but we need to find a way to find those and to target those that actually, for whatever reason, are not coming to the facilities and are not adhering to their clinic visits. And then limited use of viral load testing. Now, this sounds very dramatic, and I know South Africa is one of the first countries that rolled out viral load testing in resource-limited settings. So we are definitely in a better place than many other countries. However, we still only do 1 viral load in patients that are suppressed. Now, 1 viral load in those 12 months. So you come to the clinic today, you have a suppressed viral load. Clinician says, great, well done, keep up the good work. And two weeks later, you have a death in the family, for example. And for whatever reason, you are not doing well, you kind of not great with taking your treatment. Your next appointment is only 10 months later. So if this is a continuous thing and you don't develop any clinical symptoms that make you go to the clinic, you actually might be failing for 10 months before you have your next viral load done. Then you have a detectable viral load, but we need to have a confirmed viral load. So they need to come back a couple of weeks later. So obviously there is like a significant amount of time in between if something goes wrong, that there is a chance of developing resistance. And then finally, delayed treatment switches. So if patients are failing and the underlying issues are not addressed and the patient keeps on failing, but they are still taking the drugs, the number of mutations that are being selected and that are in the replicating population will just continue to accumulate. And the more mutations you have, the more risk for cross-resistance to any of the other available drugs from that same class. Okay. Then finally, the national program, again, gaps in ART program delivery goes again with stockout, access to viral load testing. But then also, if the country has no plan or action to actually monitor resistance, for example. So WHO recommends to do regular surveillance on drug resistance. And obviously, you also need to have a plan of action when your results come out and there is like a flag in those results. An example for that was the high level of NNRTI resistance in patients that are initiating treatment. So pre-treatment resistance, more than 10% have at least 1 NNRTI mutation. And that's the reason why now a lot of countries have switched to Dolutegravir, because too many patients we knew in advance were not going to do well on an efavirenz-based regimen. Okay. So that's it on the principles and the factors for resistance. So now I'm going to switch gears and I'm going to try to explain this in an understandable manner, how we actually do the test in the lab without going into the nitty-gritty. If you do have any questions later on, my email address is on the slides. Just drop me an email and we can chat about it. So we start from our sample, so preferably plasma, EDTA or PPT. But we can also do it from dry blood spots. So if you do have an infant of a mom that is failing a DTG-based regimen, it might be interesting to actually see if there was any resistance transmitted to the infant. And in those cases, we can use DBS. Also, we do some testing for Malawi and Swaziland, Eswatini. And they send DBSs just because it's easier to transport. You have to, however, take into account that what we call the lower limit of detection is a lot higher for DBS than for plasma. So plasma remains the preferred sample to start off from. Then we isolate the viral RNA. We then do PCR. After the PCR is done, we have to sequence and we analyze the data and we generate a report. This whole process from start to finish, if nothing goes wrong, takes at least 4 days, i.e., that is why you sometimes need to wait a bit longer for your resistance result. And because we don't get a lot of samples, we batch our samples. So we don't start a run every day when the sample comes in. Okay. So again, EDT-derived plasma, the dry blood spots, or plasma separation cards could be a possibility as well. The principle of our RNA extraction. So we have our sample with the virus. We add a lysis buffer that will break up the core component of the virus so that we have access to the nucleic acids. Then we do a number of wash steps to get rid of like all the non-nucleic acid stuff, the proteins, the lipids, and so on. And then we use some kind of affinity purification. I'm not going to go into the detail, but in the lab, we kind of have methods to actually attract the nucleic acids and to make it as pure as possible. And that is what our starting material is. Then we start with the amplification process. So this graph is the HIV genome. It's about 10,000 base pairs. And the region that we're interested in is the POL region, or the polymerase region, which consists of protease, reverse transcriptase, and integrase. The reason why we target those regions is obviously because we're using protease inhibitors, RT inhibitors, and integrase inhibitors. So in most cases, the mutations develop in the genes that translate into those enzymes. Okay. So our standard assay was protease and RT. So we would cover almost everything of protease and the first portion of RT. The reason why the last portion is not done is because there are no clinically relevant mutations known in that region. And the longer we make our fragment that we need to amplify in the lab, the harder it is to get a robust assay in the lab. So that's why we keep it as short as possible. Obviously, now with integrase inhibitors being the mainstay of the treatment, we also need to do integrase testing. And at the moment, we have to do it in 2 separate tests. Again, if we would want to do one PCR product of the whole length, we're going to have too many samples where we can't amplify and we can't issue a result. And that's why we split it into 2. Okay. So what happens then? So we start from RNA. In the test tube, we will then do what we call cDNA synthesis. So it's translated into cDNA. And then we will use primers, those little arrows, to actually multiply that cDNA. Okay, so we will then get PCR products, and the PCR products are determined by the primers that we use. So we design our primers in such a way that we cover all the relevant mutations in that specific fragment. Okay. Then we do a second PCR on top of the first PCR, and that's to make it more sensitive, especially for the ones with low viral loads. So we actually use our PCR product as our template, and then we use a different set of primers to generate our final PCR product that we're going to sequence. Okay. I'm going to skip through the technicalities. Once the PCR is done, we have our first, what we call QC check. So we run a gel in a tank like this, and the gel actually, your DNA fragments migrate through the gel, and smaller fragments will end up at the bottom, larger fragments will end up at the top. So we can distinguish the size of the fragment that we've amplified, and that is our first QC check. So here you see a good band, so this is something that we can proceed with. In lane 5, we don't see any amplification. In many cases, this is our negative control, so that's a good thing because we don't want anything in the negative control. But if this would be a patient sample, then we will repeat the extraction once, just to double check that we haven't made, like that there was no artifact why it didn't work. Okay. The reasons for no amplification is often low viral load, no recent viral load. So if you send a sample and you say, oh, the patient had a viral load of 5,000 3 months ago, and you take a sample today, that doesn't mean that the viral load is still 5,000. It might have gone up, but it also might have gone down. So that's why it's important for us that you guys provide us with recent viral load results as much as possible. If you're uncertain, just ask for a viral load and a resistance test at the same time. You don't need to wait for the patient to come back, okay? If the viral load is below 400, which is our cutoff, then we will reject the drug resistance test and there is no charge for it. So if we see that there is a viral load requested, we will always wait for the viral load results to come out before we start the process of drug resistance testing. Then we go to sequencing. So in sequencing, we do the same as what we do in PCR. So we actually also amplify the PCR product, but in this case, our building blocks, so the nucleotides, look slightly different. So in a normal nucleotide strand, you have DNTPs, okay? And they look like this, and they have an OH group here. When the strand is building, so every time you add a nucleotide, you need that OH group to make a bond with the phosphate group of the next nucleotide. I know this is a bit of chemistry, but this is how they actually attach to each other, right? When we do sequencing, we have these ones, but we also have the DDNTPs. And the difference is that there is no OH group at this position, meaning that if something like this is incorporated, the next nucleotide cannot form a bond. So it's what we call chain termination. So your whole amplification process stops, okay? These specific ones, the DDNTPs, are labeled with a fluorescent label, which means that all the strands that have been terminated have a color at the end, okay? And that's the color that we detect in our sequence. And because we know that, for example, red is a C, if we pick up a red color, we know that at that position we have a C, and that's how we determine the sequence, okay? This is like very, very brief. If anybody's interested in like the details, just let me know. So what happens? We have a double-stranded PCR product. We first do a denaturation to split it into single-stranded DNA strands. Then we have a sequencing primer, which we've designed that it will fit to this specific DNA fragment. And then as we do the extension or the actual amplification, we have a pool of our normal building blocks, the lines, and the ones that are the chain terminators, and those are fluorescently labeled. Okay, because the fluorescently labeled blocks are there in a minority. In most cases, you will have a normal building block being built in, and by chance, at different position, a terminator will be built in, and that's how we generate different size fragments. And those different size fragments are then put on this instrument. They are pushed through this kind of capillary. The principle is the same as our gel electrophoresis. So the fragments will be separated by size, so which means that the smallest fragment will actually, so this is an example of one capillary. The DNA fragment is inside, it gets pushed from one side to the other side. The smallest fragment will go fastest, so the smallest fragment will pass the laser first. So the first signal that we pick up is of the smallest fragment, and we pick up which the color is. Then the next fragment that passes is slightly larger. So the second color that we pick up is this, and that's how we actually build the sequence. Okay. So the outcome of this is a graph like this, which is what we then need to analyze. Okay. And I just want to point out that in some cases, you have two colors on top of each other. Okay, the peaks are not the same height. Going back to the viral population and having viruses that are slightly different from each other, this does not mean that in a single virus, you pick up two different nucleotides at that position. This means that some of the viruses will have a red one and some of the viruses will have a blue one, which might translate in a mixed mutation. So sometimes you see K103KN, which means that the patient has, some of the population is still the wild type, the K, and some of the viral population is N. Okay. So after we finish this, then we end up with something like this. Obviously, nobody knows what to do with this, but then we use the Stanford database, which is freely accessible and has a whole lot of information to actually put this sequence in. You can select the drugs that you want to include in your report. And what we do on TrackHere is exactly the same. So we have a link with Stanford in the back of TrackHere. So we copy paste that Chinese looking string of letters. And then TrackHere will actually automatically generate the report. So it will give us the mutations and it will give us for all the drugs, the interpretation from susceptible to high level resistance. And that's what you have access to. Okay. Now, I just want to come back to some of the quality checks that we do. So this is a PCR gel from our lab. Number 5 is no amplification. So if you get a report, no amplification obtained, this is why we give you this report. That means that there was not enough material in the sample to actually amplify the region that we want to sequence, and therefore we cannot give you a sequence. Okay. Again, often it's low viral load, poor sample storage. So if the sample comes from far away and it takes 7 days to get here, the RNA might be partially or completely degraded, and we don't have good material to start from. But we can't test that upfront. We have to do our PCR to check. In this case, you see the band that we're looking for, but you also see another band, which is smaller because it's at the lower part of the gel. But when we do the sequencing, we actually sequence both of these. And that sometimes generates what we call poor sequence quality. So sometimes you get a report saying poor sequence quality obtained. That means we had a PCR product, but the actual quality of the sequence was not good enough for us to give an accurate result. And that's why we ask for a sample. Again, this is mainly due to partial degradation. So some of the viruses are still intact, giving us the product of interest, but some of the viruses are kind of cut. And if it's cut in between the region that we actually want to sequence, then obviously we can't give you a good sequence. So this is what a good sequence looks like. So nice, distinct peaks. This is what a bad sequence looks like. So if we get something like this, obviously there is no way for us to determine what is a mixture, what is background. So this is what we call a poor sequence quality. Okay, just to put it a little bit in perspective. So in summary, if you get a no amplification result, we're not able to get a PCR product. Usually it's samples that are below the limit of detection for us. So especially for those that had a viral load that was not done on the same day. Sample integrity, and very rarely a primer mismatch. So obviously we don't have individual primers for each patient. We take a generic primer, and very rarely, but it's very hard to prove because we can't get the sequence, the primer doesn't bind. And that's why we sometimes have a backup assay with different primer sets so that we can try with a different primer set to get a result. Poor sequence quality, usually when the PCR product doesn't look great, it can be a random error, and that's why we repeat sequencing once if the initial result is not good. And again, we also use primers for sequencing so that you might also have the mismatch. Okay, so why is resistance testing important? On a population level, the surveillance is important for us to inform guidelines. The example I gave you from the switch from efavirenz to DTG-based first-line treatment. Obviously, individual patient management to identify optimal treatment for each individual, but also to reduce toxicity by replacing the inactive drugs that might have toxicity. But this option is less relevant with the safer drugs. Obviously, if you have very little side effects, then this is not as important. And also to avoid unnecessary treatment switches. In the case of DTG, if you have a patient failing a DTG-based regimen, but there are no DTG mutations, there is no reason to switch them off a DTG-based regimen, okay, unless there are side effects. So when do we do resistance testing? First of all, it's not useful to do resistance testing in patients failing NNRTI-based regimens. The reason is because the resistance profiles are extremely predictable. And in the old days, obviously it's a little bit less relevant now, switching them from an NNRTI to a PI-based regimens, it didn't matter whether there were mutations in that virus, yes or no. And that's why from an individual patient management point of view, you don't need to do resistance testing in an NNRTI-based, for NNRTI-based failures. It is useful when there is moderate level of resistance, for example, for PIs, so the Dolutegravir, atazanavir. When we did a study, which is already almost 10 years ago, we found that in patients failing PI-based regimens, only 16% had PI mutations. Now that prevalence has gone up quite a bit. I think we're now at about 30%. But doing resistance testing then is much more important because if they don't have PI mutations, there was no reason to switch them to a third-line regimen. It was more about managing toxicity, adherence, and so on. The question is, what do we do for drugs with a very high genetic barrier? And I put very in brackets because I'm not too convinced anymore that we have a drug with very high genetic barrier. Okay. Our prevalence of DTG resistance might be higher than we initially anticipated. And we need to find a balance on when to do resistance testing because we all know resistance testing is expensive. I think we currently charge 2,300 Rand for a test. At the moment, we charge double if you need integrase testing and PRT. We have validated another kit that we don't have to do the double charge anymore. but NHLS is being a bit difficult in approving it and getting it on the ordering system. So I'm working on it. Hopefully soon we will not have a difference anymore in the costing. And then maybe we should start thinking about alternative monitoring tools to actually identify patients that are at the highest risk of resistance. It's extremely difficult. Adherence measurements is like the hardest thing, and I applaud you for all of you who try to figure out if the patient is telling you the truth or not. But there are some indications that maybe drug exposure testing, so a drug level testing, it's not good for individual therapeutic drug monitoring. So don't use it to see if the patient has enough drug to actually suppress the virus. The thing is that if we do drug level testing and you have a negative result, there is no reason to do a resistance test because the patient is not taking their drugs. So the chances of having resistance is much lower. However, for those that that have detectable drug levels, there might be two groups. Patients that are taking their drugs religiously, they have mutations, and obviously it's not sufficient to suppress the virus. On the other hand, you might have patients that know what is expected from them. They're not very good at taking their treatment, but they know before they come to the clinic, oh, let me just quickly swallow a pill because that's what the doctor wants me to do. So obviously you're also going to have patients where you do find drug levels and they are not going to have resistance because they're actually not really taking their treatment. Okay. So Dolutegravir resistance. So DTG resistance in ART naive people living with HIV. So if you start a patient on TLD1, according to the guidelines, so they haven't had any previous exposure, development of resistance is extremely rare. So I think there is very little justification to do resistance testing in these patients. Okay. The challenge is that many of the patients that come as a new patient might not really be new patients, and they might have been treated at a different facility. They might have defaulted their treatment and not disclose it for whatever reason. So making the distinction between TLD1 and TLD2 is not always so easy. There are a handful of cases described in case reports, and recently there was one clinical trial where a naive participant actually developed DTG resistance. For those that are ART experienced, but DTG naive, so to DTG, if you take DTG monotherapy, there is it's very likely to develop resistance. So there have been trials done because it was such a wonder drug to see, can we just treat patients with a single drug? The answer is no. Like, just don't do it. Then there are, for those with DTG +2 NRTI, so that's what we currently use, it's highly efficacious when they have failed an NRTI-based regimen. So if a patient, and highly efficacious in terms of viral suppression. So patients that have been on TEE in the past, regardless of viral load, you can switch them to TLD. However, I would just recommend, if there are any adherence issues, please address them, because they go from one pill once a day to one other pill once a day. If they're not taking the first pill, why would they start taking the second pill? So, and I think that's one of the reasons, I think too many patients with adherence issues are actually being switched blindly to TLD without too much support. And I think that's why we're seeing more resistance than what we initially expected. And then I just come back to that thing in the guidelines about the 80%. I'm not completely convinced that that is the right thing to do. So how much resistance are we seeing? So this is just a list of some of the trials that have been done. And then the last one is an implementation program from Malawi. Graham Minky's actually best estimate 1 to 3% of patients on second-line TLD, so TLD2, will develop resistance between 6 and 3 months. So that is 1 to 2% of patients who are switched to TLD2. That is not 1 to 2% of patients who are failing TLD2. And I'll show you some data on that. So he did like a back of the envelope kind of calculation, but recently there was a paper published by Bob Schaefer's group who developed the Stanford database, and they actually looked at the literature, they looked at clinical trials, case studies, and so on, and they found 99 cases, published cases of DTG resistance. And their resistance rate calculations is less than 0.1% in ART naive, and 2.1% in those with previous NNRTI failure. So very similar as what Graham had calculated. Okay. In the same paper, they also looked at the different resistance profiles. And I'm not going through all the different mutations, but I just want to note that there are different pathways to develop resistance to the DTG. Initially, it was thought that the 263K mutation was the mutation. It does appear that's this blue block, but there are also other mutations. And these mutations are also, especially the 148 and the 155, were mutations that had been identified in first generation integrase inhibitors in the past. Okay. Then there was a recent paper in the Lancet. They looked at risk factors to develop resistance. They looked at cohorts. Unfortunately, most of the cohorts were done in the West. There was a little bit of South African data, but very limited. So just take into account that this is in a Western setting where there is very frequent viral load monitoring and mainly subtype B. They indicated that obviously monotherapy has the highest risk of developing resistance. So the more to the right, the higher the risk, and dual therapy as well, okay, which is not relevant for us because we use triple therapy. And then at the bottom, sorry, they did look here at subtype B and non-B. There is a slight difference, but as I said, they had very little non-B. So I think this needs to be reevaluated with, for example, data from us. The other thing was that the presence of NRTI resistance in the background seemed to predict a higher chance of developing DTG resistance. So if patients already have resistance from their previous regimen, the chances of developing DTG resistance is higher. So last two slides, some of our data that we presented at the end of last year. So just to put this into context, so in our lab, we only get samples from patients that are failing, first of all. So it's not the whole population. So don't compare this to the 1-2% that I mentioned earlier. Also, we are only getting samples from patients for which the clinician is suspecting resistance. So this might be a bit of an overestimate, but I'm not convinced it is really an overestimate because I think not enough patients are getting resistance at the moment, are getting resistance testing done at the moment. So we looked at the data from July 2021 to April last year. We did 162 samples and 26% of them had at least one integrase mutation and 24% of them had DTG resistance. And we defined DTG resistance as intermediate resistance up to high level resistance. Okay. Just indicating what kind of patients had testing done. So half of them had previous PI-based regimen. A quarter had previous NNRTI-based regimens. And then there was still 16% for which there was no treatment history on the form. Looking at the different mutations, the main reason, so it's not so much for the mutations themselves, it's just to let you know that there is a wide variety of mutations that we pick up. Some have only one, some have 4 or 5. I just want to point out, for those that we had duration of treatment on the form, the ones in bold are the ones that actually had resistance picked up before the 2-year cutoff that was included in the guidelines. So, and I think that 2-year cutoff is one of the things that the Clinician Society and maybe also the DOH is going to reconsider soon, because I think we had a case in Helen Joseph that developed resistance after 4 months of DTG exposure. I mean, it's anecdotal and hopefully it's not for everybody, but DTG resistance is not uncommon. That's the take-home message. Okay. Very last slide, the surveillance that we did. So we did drug resistance surveillance using leftover viral load specimens. Because we don't have treatment history for those samples, we use drug level testing as a proxy to try to identify if they were on DTG or not. However, almost half of them, we couldn't detect anything, indicating that, again, failure is still most commonly caused by poor adherence. Okay. If you look over the years, for those that we did have drug levels detected, you can see that the efavirenz levels are going down. So in line with the guidelines, fewer patients are on an efavirenz-based level. and DTG is going up, but it's still like I was expecting a much higher number. And for the 2023 survey, it hasn't gone up that much more from 15%, just slightly. What I do want to point out is that in 2019, we didn't do integrase testing because there were no patients on, or very few patients on DTG. In 2021, 0.2% with resistance, 2022, 1.6% on resistance. That was from all the samples. However, if we look at those with confirmed DTG exposure, so we do find DTG levels in the sample, it went from 2.7% in 2021 to almost 12% in 2022. Okay. I can tell you for 2023, it's not going to 22%. So it's not as steep as what it was. But when we saw this, so again, patients that have confirmed DTG exposure, and that's where I think this drug level test might come in handy. Those ones are the ones at highest risk for DTG resistance. So if we need to triage, this test might be able to help us to just check out the ones that are not taking it anyway in an objective manner by doing drug level testing and doing resistance testing on those that do have DTG levels. And that was my very last slide.